Vicki is, as a professor of neurology, she leads the Movement Disorders program at the University of California at Davis. And she's going to talk about a particular brain disease, Huntington's disease, a genetic disease, and that profoundly affects the brain. And she's going to talk about some amazing new technologies and possibilities for therapy that are based on some of these new advances in neuroscience.
So,
if we can get your computer working.
that's right. If the computer will connect up here. So thank you so much. It's a pleasure to see everyone. You know what? There's seats up front here.
Yeah.
Lots of empty seats for people who don't want to hold up the wall back there.
Yeah.
And it would be nice for that
Sit next to you,
PowerPoint
Mother.
to show up. Let's see. Oh, beautiful. Okay. Very good. Thank you. Okay. Oh, that's going to be okay. And actually, I'm going to stick by the keyboard. I was just telling Dr. DeCarli, I don't talk with my hands as much as he does. I don't have quite that Italian heritage that he does. So anyway, thank you so much for coming. And I'm going to talk a little bit about Huntington's disease and about the progress in stem cell research and how we hope that stem cell research will lead to new treatments for neurological diseases. And we happen to be a little bit further ahead on Huntington's disease than some other neurodegenerative diseases, but what we're learning doing this we think will have wide applications to other neurological diseases. So, I'm going to briefly tell you what is Huntington's disease. Some of you in this room have a lot of knowledge and information. Others might be new to it. I'm going to talk a little bit about basics of stem cells. We'll talk about the particular type of adult stem cell that we're using in our research and give you an update about progress in a grant that we received 2 years ago to develop this treatment to take forward, hopefully into patients in a phase 1 safety and tolerability trial. So Huntington's disease is a slowly progressive and sadly fatal neurodegenerative disease. And actually, I've changed my slides the last few years to call it a neuropsychiatric disease. The longer that I've been involved in working with patients with Huntington's, the more apparent it is that the circuits that are affected in Huntington's disease very strongly affect people's cognition and behavior and emotions, and probably those symptoms might be even the worst symptoms with this disease. It's a very rare disease. There's only about 30,000 people with Huntington's disease in the United States. There are probably 5 million with Alzheimer's disease. So we're talking about a small disease. Because it's hereditary, though, there's a pool of people who are at risk of developing Huntington's, and that's 5 times as many people, or about 150,000. The age of onset is as low as 2 years and as high as 80 plus years, but the average person with Huntington's develops the symptoms in their 30s and 40s. We estimate that the cost of this disease, although it's a relatively small population of people, about $2.5 billion per year, because people who get Huntington's can't work anymore. They can't support their families. They require caregiving, and they require a lot more and more medical care, so it has a very high impact. And the symptoms of Huntington's are 3 different categories: involuntary movements, psychiatric and behavioral changes, and cognitive loss. So this slide is showing a pathological section. It's straight down the brain like this, and the image there under unaffected would be someone who's died and had their brain examined after death, no Huntington's disease. And the one on the right is someone who's died with very, very advanced Huntington's disease. And you can immediately see there's a difference between those 2 brains. And I've circled there in red the striatum. The striatum is a deep area of the brain that Dr. DeCarli didn't spend a lot of time talking about, but it's an area where there's a collection of nerve cells that are involved in these networks and circuits that modulate movement and thinking and feeling and emotions and cognition. And that's the area that's selectively vulnerable with Huntington's disease. But as the disease progresses, there's more widespread changes that happen, because as those networks and circuits start to fail, other cascading areas begin to fail. So I've listed here the movement symptoms. The most well-known is chorea, which comes from the Greek word for dancing. So people have dance-like movements that are involuntary throughout their body. But they also have a lot of other motor symptoms there: changes in balance, loss of fine motor coordination, difficulty swallowing, difficulty moving their eyes. And then here I've listed the common psychiatric and behavioral features of Huntington's disease that happen along with the motor changes. Sometimes they precede the motor changes, and you can see that people can have depression, anxiety, OCD. They can have difficulty with multitasking. They can have difficulty with planning organizing, starting and stopping activities, a wide array of behavioral symptoms. So, tragically, Huntington's disease passes from generation to generation. And the gene for Huntington's disease was discovered in 1993, 22 years ago. And I think 22 years ago, many of us thought there will be a cure around the corner. Here's the gene. We know exactly what causes it, which is very, very distinct when we think about most other neurological diseases and most other medical diseases. We don't know the specific cause. And I think that's why we are where we are with Huntington's, is because we have a very powerful biological target. This is where the problem is that causes the disease. Every person who has Huntington's disease has a chance of passing it on to their children, a 50-50 chance. If you have a parent with Huntington's disease, you have a 50-50 chance of inheriting the disease. Each child is like a roll of the dice, has that 50-50 chance. The mutation is actually an expansion and a stretch of the gene that has the message CAG in the genetic code. There's an expansion of too many of those in a row, and if there's more than 35 CAGs in a row, that makes this gene and this protein toxic, and it causes brain dysfunction. If it's low, if it's less than 27, there's no change that occurs with this. So how does that mutant Huntington protein cause brain dysfunction? It's actually present in every cell of the body, but as far as we know, the symptoms are restricted to parts of the brain. And we know that with Huntington's disease, the mutated protein builds up in cells, and the cells are the medium spiny neurons in the striatum, in that area that I circled. That's where the pathology starts. And we see clumps of protein that's misfolded, that can't be broken down, that can't be packaged and handled by the cell in the way that other proteins normally are. And this is actually a theme in neurodegeneration. Each disease has its own clump of protein that builds up, whether it's the plaques and tangles in Alzheimer's disease or a number of other things in other degenerative diseases. We still, 22 years now, don't know the exact function of the Huntington protein. That's kind of a big mystery. There's a lot of areas where it's affecting function, but we do have a very strong biological signal that there's a trophic factor, a growth factor, or a brain fertilizer, if you will, that is very necessary to maintain the health of brain cells, of neurons in the brain, and also to kind of help keep new cells forming, because we do have some stem cells in all the organs of our body, including our brains, that kind of replenish. And this factor is called BDNF. It's brain-derived neurotrophic factor. That's the molecule. It's big and complicated, and that's the shape of it. And the levels of that protein are markedly reduced. We think that the Huntington mutant protein blocks the formation of BDNF, and it also blocks the transportation from the outer part of the brain into the striatum. Without having BDNF, those striatal neurons will die. So at our Center of Excellence, which is on the other side of the causeway, we have a clinic. And our clinic was started because of families in the Sacramento and Davis area who approached our department years ago asking for help with Huntington's disease. And they have actually funded the people that you see in these slides who come and help take care of patients with Huntington's. And I couldn't be prouder of the team that we have of people who are helping to treat people with Huntington's, and we give them every treatment we possibly can. We help treat the psychiatric symptoms, we help them with motor symptoms, we help them with their cognitive symptoms, we do education, we do outreach, we do genetic testing. At the end of the day, though, we still don't have an effective treatment to slow or to halt Huntington's disease. We also do a great deal of expert education to people, other health providers, to a number of people who are involved in our society in supporting people with Huntington's disease. But one of our major objectives in our center is to advance Huntington's research. We've been doing research in Huntington's disease now for 18 years through a consortium called the Huntington Study Group, and we're still working with them. But because of what we've learned with that, we've been able to parlay that into something very unique and special here at UC Davis, where we are known throughout the world for doing our Huntington's research. In fact, I'm told that people who either are across the Pacific Ocean or across the Atlantic Ocean call us Yuck Davis. They kind of know there's something going on at that Yuck Davis place that has to do with Huntington. You should contact us. So back in 2009, we formed a new partnership with our stem cell director. The California Institute of Regenerative Medicine was formed in 2004. voted for in 2004. 2007, our stem cell director, Dr. Nolta, came to UC Davis, and I met with her and told her, stem cell, I don't know stem cells, but I really need your help. Is there anything you could do that might help our families who have Huntington's disease? And she was very polite and very kind. She had a big center to set up and build, but actually it's when she met the families affected by Huntington's that she really became very impassioned about helping people with Huntington's. And so I'm standing here representing her work and our partnership. I'm a neurologist, she's a scientist, but together we've been able to forge a partnership that's helped out with this. And we actually do the work at the Institute for Regenerative Cures, which is back over across the causeway in Sacramento. A great, great institute. I could tell you lots about it, but I don't have time. Well, there's 200 scientists working there, and it's 100,000 square feet of research space, and they're working on every disease in the body. There you go. And Huntington's disease. So if you could go inside of that building, you would see this is the Good Manufacturing Practice Facility. It's an amazing, not just state-of-the-art, but state-of-the-planet facility. There are things there that don't exist anywhere else on the planet that were designed by Dr. Gerhard Bauer, who is the director of the GMP facility, and a tremendous amount of stem cell research is going on in that building, and they're also using that facility to support other institutions who are doing stem cell research. And we're hoping to have a treatment in that facility for our patients. That's what we're working toward. So let me change directions now and talk about what is a stem cell. So a stem cell is a single cell that has specific properties that are very special properties. One is that it can replicate itself, and it can replicate itself over and over and over again in multiple passages and restore and replenish so that there can be a constant supply of stem cells. Another thing that's special about stem cells is that they can turn into other tissues. And there are different types of stem cells. Some have more ability to do this than others, but this is why we're excited about stem cell research. There are two basic types of stem cells. One are the adult stem cells that we're working with in this project. The other are the pluripotent cells. These are the cells that can truly turn into multiple different tissues anywhere in the body. And these are both the embryonic stem cells that are derived from unused embryos from in vitro fertilization, and also induced pluripotent cells that are engineered from donated skin cells. There's a way to get in there and engineer them back into the state of being pluripotent. Mesenchymal stem cells are very special cells, and I'm beginning to appreciate more and more and more just how special they are. These are bone marrow derived, but they could come from belly fat. There's other places in the body that they exist, and they have a number of important properties, and I put little arrows on those important properties. One thing that they do all by themselves is to secrete trophic factors, including BDNF and other factors that help to support brain function. They help out with blood supply. They help out with the synapses talking to each other. They just do that on their own. They help with neurogenesis. They help to create an environment where new cells can be formed to help replenish sick and dying cells. They reduce inflammation, which is an important part of cell death in a lot of diseases, including Huntington's disease. So back in 2012, Dr. Nolta's team wrote a paper that talked about her brilliant idea, which was engineering these mesenchymal stem cells as a therapeutic to treat Huntington's disease. So these are not cells that normally occur in the brain. These are cells that occur in other parts of the body, but they have these special properties, and she's a world expert on using these cells and engineering them for other therapies. So the idea is this. If this is a brain section, sort of a cross-section of the brain, if you look on the outside, the cortical neurons are on the surface of the brain, and they have connections that they're sending down to the purple cells, which are the striatal cells, the medium spiny neurons, and they're constantly delivering BDNF. That's the normal state. That's what we see in people who don't have a Huntington mutation. In the presence of the mutant Huntington protein, that delivery of BDNF is stopped, is blocked, and the cells die out. And the idea is to load up the MSCs in green here with little red dots of BDNF and deliver them to the brain, and that will then restore the medium spiny neurons. Those that have already died will not come back to life, but those that are sick can be improved, and new cells can be recruited through neurogenesis. That's what the brilliant idea is that Dr. Nolta had. So in 2011, we submitted a grant to our state stem cell agency, and we were very excited when the grant was approved. It was a day of great celebration. This was the actual day when the committee voted to fund our grant, and there are members of the Huntington's community here in the picture who were there that historic day to speak at the microphone about how important this research was to them. They're always there. There are always people from the Huntington's community there letting CIRM know how important this is. And there was much confetti that was released because it was truly a historic day in Huntington's research. So this is the name of our grant, mesenchymal stem cells, these adult stem cells, engineered to produce BDNF as a potential treatment for Huntington's disease. The objectives of our grant, number one, are to obtain the approval of the FDA to go forward with these cells in a 1 to 2 year treatment trial that will look at safety and tolerability. And the second bullet point, what our gene therapy candidate is, safety modified, donor derived, human mesenchymal stem cells engineered to oversecrete BDNF as a neuroprotective strategy to help slow down the progression of Huntington's disease. This is where I tell you and I tell our patients, this is not a grant to cure Huntington's disease. This is an important step, we hope, working toward that. And there are people in this room, I'm going to call out Dr. Kyle Fink and his team in the back there, who are working on amazing techniques to edit the gene and correct the mutation. But we're not there yet. We're using this grant to work out how to engineer those cells, how to make them safe, how to make them work, and to test them in people. Future trials, we hope, will lead to something that could potentially be curative. So this is the project plan. When we wrote the grant, we made a plan that would be, first of all, it's a 4-year grant, and then the money ends. So we have a very ambitious grant here. In the first 2 years, we're working on all the preclinical studies in the stem cell lab. The team is working night and day, doing all types of experiments with these cells, getting them ready, testing them in mouse models. But during this first and second year, we've actually started an observational study called PreCell. And PreCell is a study for people who think they might want to be in the future stem cell trial, and they agreed to come into the study. There's no treatment. All we do is have them come for 2 days every 6 months, and we're measuring how are you doing, how is your Huntington's doing. We're doing neurologic tests, psychiatric tests, cognitive tests. We're talking to their care partner. We're giving them surveys about their mood and their quality of life. We're collecting blood specimens. We're collecting spinal fluid that bathes the brain so we can measure BDNF and measure other things. And we're doing brain scans in that study. And our hope then, and by the way, I think this is going to move over. We are right here. We are just about halfway through this grant now. So we're moving into when we plan to start the treatment trial, which we're calling HD Cell, for the HD cellular therapy trial. And it's our hope that at the end of this year or the first quarter of 2016, we'll be ready to go into patients. I do have to say, we may not get all the way there, so we're working very hard to do that, but there are some lines of regulatory approval that we're currently working on to get us all the way there. The FDA has to license us to be able to do this. The Institutional Review Board at UC Davis has to give us their approval. We have to make sure that our plan of doing this is safe. We're primarily invested in safety and tolerability of this treatment, and so we have a lot of hoops that we still have to jump over. So the idea is to take these donor-derived cells from people who don't have the Huntington's mutation, bring them to the lab, culture them, grow them, store them, engineer them, and ultimately deliver them to patients. This is a beautiful slide just showing the MSCs. They are housed in flasks. I think they have apartment blocks for the MSCs in the stem cell lab. So if you went into one of the apartments, you'd see in the flask all these beautiful cells. And these cells have been engineered by adding some DNA to them to get them to pump out BDNF. And do they make BDNF? So if you look at this graph here, it's showing the wild type WT is over on the left. That's the normal small amount of BDNF that they make. But the team's been able to engineer these cells to really produce high amounts of BDNF. That's what our goal is. And we're looking at the amount of BDNF to figure out how many cells to put in the patients based on how much BDNF they are expressing. I'm going to turn now and tell you a little bit about the work in the preclinical work in the stem cell lab that involves using mouse models. One of the major advantages in Huntington's disease is that we can take the human Huntington gene and place it into a mouse, and there's different types of models of this to model the disease, and then we can test out treatments in the mouse model so we can learn as much as we possibly can from that model before we go into humans. So there are 2 models that we're using. One is called the YAC128 mouse model that has the full-length mutant Huntington protein from humans, and the R62 mouse has a fragment of that Huntington's gene. The YAC128 mouse model is similar to the human model in that as they get older, they start to have changes in their coordination and in their behavior, and their brains start to shrink in the same area that human brains do. The R62 model is a much more toxic model where the mice at a young age start to show a lot of neurological degeneration and seizures, and they actually die early. So it's more of a model of juvenile Huntington's disease. And we're testing these cells, both the MSCs and the engineered cells, in 2 mouse models so we can learn as much as we can about how effective it might be and how safe it is. I wanted to show you here that this is actually from the lab from Dr. Kim, who was a stem cell scholar who worked in the lab 3 years ago. She took a snapshot when she was in there. This is a wild-type mouse, and it's in its little apartment, so it has this bedding that's put in there every day, and the wild-type mouse is busy. It makes itself a nice, neat little bed over there. The Huntington mouse, not so much. The Huntington mouse can't figure out how to make that nice little bed and can't really get organized and just can't take care of itself. And it's a simplistic idea, but those mice that have the Huntington gene have abnormal motor behavior and abnormal thinking, and they model this disease for us in a very special way. So the engineered human stem cells, which are green in this slide, are actually implanted in the mouse brain. The mouse brain cells are dark blue on this slide. And you can see that the human MSCs making BDNF, the green ones are there, present on this higher view. And then each transplanted human cell is giving off BDNF to deliver it to this area of the brain. And that's the model of looking for the potential benefit of these cells in humans. A lot of studies are going on, many studies are going on. I just brought one example. These are happy, sleeping mice. They're alive, okay? They're in a little special camera. They have anesthesia there, so they're sleeping. And they've been given stem cells with a special tag on them so that you can look at those mice without doing surgery, without going in them, just on the outside and look at the signal from those cells that enables the team to see how long are those human stem cells surviving in the patients who have, in the mouse patients who are the YAC128 mouse model. And they've done tremendous work to engineer the system so that those cells will live long enough to do what we need them to do. And this is just a beautiful slide using a CT scan of the mouse brain that's been stained to show the neurons that are the bright white neurons there. And we're going to be measuring those neurons and counting them to see how this treatment is working. Here are some behavioral studies that were done, some pilot studies from a little bit ago, showing how the mice are moving around, So this is actually the wild-type mouse has a lot of rearing and exploratory behaviors and moving around. That's right here. The Huntington mouse is kind of afraid and they can't move very much. When these special stem cells were put in, the mice treated with these special stem cells were more like the wild-type mouse, so it showed some improvement. Same thing over here for moving around. And there's another study that showed that if you look at the number of cells in the striatum, that they were very much plumped up in this pilot study, trying out these cells in mouse models. So right now our initial data looks very good. We're still putting together all of the final data for the package to go forward to the FDA, but we're very optimistic about this. And again, I salute the lab team for the tremendous amount of work that they've been doing. I'm going to, in the last couple minutes here, update you about what's going on in the human trial. I've already mentioned pre-cell, which is going on right now, and the goal of that was to enroll a group of patients who would be candidates to go forward into the actual stem cell treatment, which we're calling HD cell. So in the pre-cell study, patients are having all these different measures taken, MRI measures, cognitive tests, we're collecting spinal fluid. We're doing this every 6 months. And we're tracking how each person is doing. We're tracking how their movement's doing, how their thinking's doing, how their mood's doing, and how their brain scans are looking. Our first patient was screened a year and a half ago. We've screened 35 patients now and enrolled 26. They're doing all of these tests every 2 months. Every 6 months for 2 days at a time. This is actually some data that's come out of the DeCarli lab. Dr. DeCarli is one of our closest co-investigators and working very closely on this project. And what he was able to do was to take some of the images from people in pre-cell and then to manipulate those images and take some measurements. And the areas that you see in blue are from the Huntington's patients. The blue areas, the cooler colors, are where cells have already died out. This is when they first come into the study, early stage Huntington's disease. We can already pick out loss of brain cells that has happened from the Huntington's mutation. That's the target. That's where we're going to put the stem cells that we hope will help to restore and slow down the progress of the neurodegeneration. So going forward, we're still enrolling in pre-cell for about 2 more months. We're completing all of the efficacy and safety studies in the mouse model, and we're preparing this year to submit our IND to the FDA in hopes of being approved to go forward into HD cell. HD cell will be a neurosurgical treatment trial. We're going to have to put the cells straight into the brain. So it's an invasive delivery system. We can't put them intravenously. We can't put them in little pumps. There's no other way to get them there. But we're using the same techniques that are used actually for deep brain stimulator implantation. So the same type of a technique will be used. Over 100,000 people have had this surgery done. It's a very safe procedure, and we'll be watching those patients very closely afterwards. This is the target area. That's the striatum where we'll be implanting those cells. So I want to finish up by thanking the team at the Institute for Regenerative Cures, thanking CIRM for the funding, thanking the patient, families, and advocates who have made all of this work possible. The red circles show you all the people there who are working on the Huntington's project. It's a major effort in the lab. And this is the last slide just showing the people here at UC Davis who are working on this project, and we have collaborators from around the world that have advised us and are actively working with us on doing the best science we can possibly do to go forward. And that's my talk. Thank you very much.